We've been discussing the cytoskeleton, and then just as a refresher, we talked about the actin and microtubule network. We've talked about the fact that how these two networks are, similar and different. Similar in the way they are put together by subunits, different because of the kind of subunits that are coming together. Similar because, they are both requiring energy. One requires GTP, one requires ATP. Different because of the way they are organized, and this picture captures that, right? The fact that these are microtubules are radiating from a central point inside the cell and appear as big long strands. These are kind of highways that are running from one end of the city to the other. And their intent is to kind of take you from one point to the other. The microtubules, along with the centrosome, also I mentioned, has a tip and a tip complex that plays a vital role in anchoring it to very specific sites inside the cell. The actin cytoskeleton, on the contrary, is more branched. It has the capability of making these very well-defined branches, which the microtubule network doesn't. The actin cytoskeleton, therefore, plays an important role in holding up the membrane, in talking to the plasma membrane. So there is something called a cortex that actually sits below the membrane and kind of holds it up. Any type of force generation in the cell, right, like pushing of the membrane, moving the cell forward, bending the membrane, will all require the actin cytoskeleton. The microtubules, on the contrary, act as major trafficking or movement highways. It's not like actin cannot allow for movement to take place. Microtubules, because they originate from the center of the cell and move all the way to the periphery, can be used to carry things from organelles like the Golgi that kick off at the center, that are present around the center, and take things all the way to the periphery of the cell. Right. So that's among the distinctions that exist. We spoke about the fact that this assembly happens through defined subunits that can be put together in various forms. And I gave you this example of Lego. And for some of you who may have not used or seen a Lego building block, they look like this, and they come in very specific defined shapes, and they can be assembled into various structures depending upon what you're trying to build, right? And that's kind of what the cytoskeleton is also like. There are a couple of movies we'll look at, and these are just refreshers on how how these are put together, how the cytoskeleton is put together. One is a very nice movie that's from the Mechanobiology Institute that shows you how actin is assembled. As I said, you don't have to remember all the players. You don't have to know, what each component is. The critical thing is for you to conceptually understand how energy requirement is there and how these structures are assembled and disassembled. Right, and that's the take home from the movie. So the movie does not have a commentary, so we'll watch the movie first. That's a single actin subunit, which is globular actin, which it's called G-actin, right? And that's the architecture of that. This is the ATP molecule that we spoke about, and which can come and bind actin and convert into ATP bound actin, which now then has the ability to assemble, right? So then there is hydrolysis of ATP to make it ADP bound actin, and that cycle of ATP to ADP is what's vital. There is something called spontaneous nucleation for the yellow ATP bound actins, where they can come together and sit together, right? And when they sit together, they start building a strand, right? And then there is hydrolysis. So you can see the ATP bound actin getting converted to ADP, and then now the ADP strand starts disassembling, and there are proteins that are required for both parts of the process. So there is polymerization and depolymerization, both happening in the same strand, and the rate at which both happen is going to determine the length of the strand, for example. A number of proteins, again, as I said, you don't have to worry about names and such. This is just to know that there are proteins that are regulating these processes, right? So polymerization as well as depolymerization. Profilin, for example, inhibits nucleation. That means when the actin is bound with it, it will not do this spontaneous making of or initiation of a strand, right? Which will happen otherwise. There are other proteins that similarly regulate actin as well. And cofilin is one of them, and we'll look at that as well. So as I said, accessory proteins regulate the dynamics, right? So not only is, because this is a dynamic process, you have to remember that things happen at a certain rate, and the rate at which assembly and disassembly is happening is going to determine, how stable the cytoskeletal strand is, and how long it stays the way it stays. Right. And so there are proteins that are mediating all aspects of this, right? Proteins that chop off actin. So there is an actin severing protein, proteins that regulate polymerization, the rate at which extension happens, proteins that regulate depolymerization. And this happens also for tubulin, by the way, not just actin, right? So the take home from all of this for us is that along with the cytoskeletal components being structures that are assembled this way, using Lego blocks, there are components that come to also regulate the rate at which the Lego blocks are being assembled, right? And the rate at which they are being taken out. And that plays a vital role in determining what the final architecture looks like, how long a strand of actin or microtubule is stable, where it goes and where it is retracted. All of this is determined by this dynamics. Last time somebody asked about the importance of dynamics. That's how important dynamics here is, that because everything in the cell is very dynamic. And I'm going to show you a movie where the actin has been labeled by fluorescent actin, right? So the actin strands are present in these cells, and we put a very small amount of fluorescent actin, which when I say fluorescent, will kind of emit light. And this fluorescent actin will go and incorporate into the endogenous actin. So you get to see what the actin that is present inside the cell is actively doing as the cell is, all the cell is doing is sitting and trying to move. And this is the front of the cell, something that's called the lamellipodium. And you can see these waves of actin as they go to the front and come back. It is literally like a wave at the ocean, right? And this is a real cell, and we are looking at actin inside the cell. So first take a look at the movie and see what all you see, right? You can see how actin at the periphery, at the edge of the cell, the content is very high. You can see how it is, it is being disassembled and is being carried back into the cell, right? And this actin at the front looks like that mesh I told you about, right? Here, this mesh, right? And it's there is a membrane that's in the front, and it's kind of actively pushing on the membrane. And that's what we saw here too, right? The front of the cell, the actin that you see in red is kind of actively pushing. Can you see that the front has moved forward in the time that the movie has been playing? And microtubules are poking through, right? These are the highways that are coming right up to the front of the cell. They are carrying things that are required for the front of the cell to do what it is doing as well. So this is the kind of combination of actin and microtubules that works together. The branching of the microtubule that we just saw here, of the actin that we saw here, is made by this protein called the ARP complex, ARP23 complex. And it binds actin and creates a starting point for a new strand at a very specific 90-degree angle. So all the branching that you see in actin will happen at a very specific angle. And the reason that it happens as a specific angle is there is a protein here that sits and binds to this actin that will allow for a new strand to form only at a very specific angle. Okay. And this branching is a big distinction between actin and microtubules. And this is something that only now we are starting to see one or two cases where there is some thought to suggest that maybe, just maybe, microtubules under certain circumstances may also branch. But for the most part, actin branches extensively, microtubules don't. And actin, because of the branching, is able to generate force in a way. It's like spreading your fingers and being able to do this, right? Versus using a single strand of microtubule and trying to push on anything, right? So this is able to generate force in a way that this will not. Okay, and that's one major distinction. I spoke to you about the fact that actin can be assembled just as microtubules can be into very different looking structures. And one of those structures is this philopodia, which is essentially the gray line that you see on the outside is the membrane. So the membrane is covering the philopodia, and the philopodia is essentially like a bar of actin that kind of pushes on the membrane and creates a very nice structure like this. So you can see how the actin comes together to create that kind of a philopodia-like structure. And all of this is mediated by the proteins that we saw that regulate the assembly and disassembly of microtubules, right? So of actin. So these actin and microtubules can, with the help of these accessory proteins, be made to do very different things, right? And that's an example of how this structure is assembled. And you can see, again, very distinct proteins, a tip complex, facin, all coming together to make that philopodia-like structure, right? It's also interesting that these actin structures are, exploited by bacteria. And this is a good example of a bacterium called Listeria. What you can see is Listeria that has infected a mammalian cell. And it uses the actin that is present in the mammalian cell to make nice tails of actin. So the bacteria has a mechanism, has proteins that will allow you to take the actin that is present in the cell in which it is sitting, and now build these tails which allow the bacteria to move, right? So the bacteria by itself does not have this tail till it enters the cell, and now it has the ability to move around. So if you see these black dots, the black dots are the bacteria, and the fuzzy tail that you see is the tail of actin that now allows the bacteria to move. So there are very interesting examples of how the coming together, the fact that this actin is present in eukaryotic cells, the prokaryotic cells that are entering the cell have also found a way to exploit and use these cytoskeletal components. Another example of how the same cytoskeletal component can be repurposed or used very differently is the example of microtubules being present in cilia and flagella, right? Which are very thick structures that move around and kind of allow cells in our body, for example, the esophageal lining cells. There are many places where microvilli-like structure or cilia and flagella-like structures are present, right? And so this is largely put together by the same microtubules, but in a cell, you saw the microtubule starting from the center and radiating outwards, right? Here, they are assembled into a ring like this. So this is a pair of two microtubule strands, 1, 2, 3, 4, 5, 6, 7, 8, 9 strands, and 2 in the center. And they are all held together by motor proteins that bind to one strand of actin and bind to the other, and vice versa. So these two strands, if my hands are the two strands, the fingers are the two motor proteins, and they are linked this way. And these motor proteins, all they do is, because they have the ability to walk on these microtubule strands, they walk, right? So they do this. This one does this, right? And when they both do this, right, in a very specific manner, these strands are getting moved accordingly. And as a result, the entire structure moves, right? And you're able to create this movement of, cilia and flagella that we all know of. The reason to highlight this is it's interesting that the same cytoskeletal component is used in very different ways. right? Microtubules also play an important role when the cell divides. And when we look at cell division, we'll look at this briefly to kind of pull the chromosomes apart. The chromosomes will arrange around the center of the cell, and then will get pulled towards the two corners. And the two corners are essentially the centrosome. So the centrosome that's in the center of the cell will divide, make two centrosomes, and now you will have an array of microtubules from both of these. They are essentially doing what they do in a normal cell. But now the tip of the microtubule, instead of going and attaching to the plasma membrane and other places, attaches to the chromosome. At a site or a place in the center of the chromosome, and that allows the two chromosomes to be pulled apart. And the way the pulling apart happens is because this is microtubule, right? And this microtubules have the ability to polymerize and depolymerize. When they depolymerize, they will shrink. So they just use the mechanism that they already have. That is, they can grow or they can shrink, and they decide to shrink. And when they shrink, they apply force because they start pulling these apart. And that's how the chromosomes get separated. So the microtubules that act as a highway in these cells that are normally growing, when the cell has to divide, now act as a way to kind of pull apart chromosomes as well. And that's really remarkable, right? It's essentially what we talked about last time. One player playing multiple roles, an all-rounder, somebody who can be a batsman if required, a bowler if required, and a wicket keeper if required, right? And that kind of diversity in function, these cytoskeletal components have. So depending upon where they are doing what they are doing, their role and their architecture could change. And this may have been a very strong reason for these cytoskeletal components to have to have survived through evolution, and be part of cells, because they have this ability to repurpose, right? To go from doing this to that to this whenever required, right? And this is again the example of the strands moving around with actin, and I just showed you what that would look like earlier. Again, this is an ATP dependent process, and so we have microtubules, actin, and we have the intermediate filaments that we wanted to quickly look at. The other interesting point that I wanted to mention is that because we talked about membrane and the cytoskeleton being present underneath, remember on the outside of the cell there is something called the extracellular matrix, and this is again a mesh, right? So that mesh is made up of proteins that are secreted by cells. We are not talking about the extracellular matrix, in these set of lectures, but at a later point you will run into them. And so when you imagine the membrane and you imagine the mesh of actin that is right underneath the membrane, you also imagine a mesh that is on the outside. Right. So there is a mesh on the outside, there is a mesh on the inside, and a membrane that's in between. And that's generally the architecture with which cells are sitting. There are places where cells are sitting next to other cells and the membranes are actually attached to each other, and that happens, in which case there won't be an extracellular matrix. But for most other places, you have the matrix mesh on the outside, you have the membrane, and you have the actin mesh on the inside. And that's the image of the cell that I want you to carry with you, right? We come to the microfilaments at the end, which are again, uniquely different from actin and microtubules. And we talked about actin microtubules, how they are similar, how they are different. Let's look at the microfilaments and see how they are similar or different. So first thing, they are the thinnest class of cytoskeletal fibers and are solid rods of globular protein actin. So the subunit that is used to make microfilaments is effectively actin, but it's assembled and put together in a very different way, right? Again, this idea of the same player being somebody else, right? And this might be one more reason why actin is still around, because not only can it do all those branches and other things, it also has the capability to form these very robust microfilament structures, right? They are designed to resist tension. So these are among the those structures in the cellular cytoskeleton that can take the most amount of pressure. So they don't buckle very easily, they don't break or bend very easily, right? And because of that, they provide a certain stability to the cell in places where that kind of stability is very important. So, actin microtubules, very dynamic, and the dynamicity is very vital to their function, but not everything needs to be dynamic in a cell. And there are places where, the cell wanted a cytoskeleton that's not dynamic. And, remarkably, the same actin now assembles into a structure that, is very different. Right. So with other proteins, they form a three-dimensional network just, inside the plasma membrane as well. So among the unique features that when compared to actin and tubulin, there is a great sequence variation within the intermediate filament genes and the proteins that they produce. This yields a great diversity in the type of structures they form. So they could form things like hair, nails, feathers, horns, are all examples of these intermediate filament protein. Keratin is therefore a good example of an intermediate filament protein, right? Fungi and plants lack these filaments, and insects have only one class. It seems like they are that as the metazoan lineage evolved, more intricate and complex tissues and organs evolved. Intermediate filaments were adapted and modified for tissue or cell-specific purposes. So when you are trying to build bigger and complex structures, this stability became more and more important. So to begin with, you may have had eukaryotic cells that lived by themselves, and as they started making complex structures, you now have the ability, you needed these kind of intermediate filaments. Among the properties of intermediate filaments that really sets them apart is one, these intermediate filaments lack polarity. Okay, so unlike actin and microtubules that are very clearly defined as plus and minus ends, these don't carry that kind of polarity. They don't have polymerization at one end, depolymerization at the other end. Okay, they have high tensile strength and are resistant to compression, twisting, and bending forces. So so they really have this ability to withstand pressure in a way that conventional cytoskeletal proteins don't. They have slower filament dynamics. And as I said, unlike actin and microtubules, intermediate filaments, are much more stable. Their intent is not to form and deform very quickly. Their intent is to form and stay formed for long periods of time. And so their filament dynamics are very different. They also interestingly lack motor proteins. Okay, and this is another important aspect of how these structures have evolved. Right, so you have actin, you have microtubules, all the things that they are capable of doing, what kind of dynamicity and the fact that they can have motor proteins that can move up and down, bind and bring these two structures together. All of that happens there. Intermediate filaments is all about stability. I form, I sit, and I hold my place. Right. And so it's not something that if it's not dynamic, chances are motor proteins that go up and down very quickly, do rapid changes, doesn't make sense. So they formed, intermediate filaments were formed without the presence of any specific motor protein. So, right, see, this is an example of how they can be assembled into coiled structures that are, it's my favorite example is of, these big twines that are made out of coir, right? And these big threads, which are essentially, individual coils that are kind of assembled into to create a big strand. And that's, what the intermediate filaments look like. And there are many of them that are present in cells, and a lot of them, for example, are present in the nucleus. This is at the at the nuclear membrane, and a bunch of intermediate filaments like lamins, for example, are present lining the nuclear membrane. So they're just as actin is present below the plasma membrane, these are present below the nuclear membrane in cells. There is one slide that I'm missing, and which is a listing. Okay, I'll bring that up later. I'll try and share it with you guys. Because we talked about the evolution of the cytoskeleton, I normally don't share these slides here, but I'm just going to quickly show you something here which tells you how these have evolved, right? And you remember I mentioned how prokaryotes have something that is similar to tubulin. This is that molecule called FTSZ, right? Which is what Gayatri's lab studies here. And it's kind of an ancestor of tubulin. right? Now the strand that it makes has a bit of polarity, but is, you know, like actin is a strand that is, you know, two strands that are wrapped around each other. Tubulin that you know of in the mammalian cells, you know, is like a hollow tube, right? So there is a version of tubulin that existed in prokaryotes, which looks very different, has some similarity structurally to the tubulin that we see here, right? And you can see the structure of the two proteins and you can see how similar they are. But the way they have assembled, you know, how they're put together, very different, right? Same for a protein called MREB, which is thought to be the kind of original actin, right? And MREB and you know, is again similar in structure to actin. It's again assembled as a strand that is, you know, two strands that are wrapped around each other, and that's very similar to the actin that we know of today, right? And it's interesting that these molecules existed in some form then, and then, you know, somehow adapted, evolved to create the complexity that exists in the tubulin strands that we know of today. This is a very beautiful table, and don't worry about all the complexity here. All I want you to see is that wherever the dots are red, right, it says that that particular molecule existed. And look at where the MREB and FTSB, FTSZ are present, right? And you can see all the early eubacteria, and to some extent the archaea have FTSZ. You know, there are some archaea which have an MREB version. They were all present in the early stages. The intermediate filaments like lamins, for example, completely missing in those early organisms, right? Actin, AR, R2, 3, all missing. Tubulin, all missing, right? Motor proteins, completely missing. Okay. And then there is a point where all of this kicks in, right? You have now alpha and beta tubulin, you have actin, you have the R2-3 complex. When actin is present in the form of actin in the early eukaryotes, you know, the branching is already in place. You know, it somehow you know, existed with that branching as part of the of its architecture. The motor proteins are in, right? So you can see the bottom blues, kinesins, dynein, myosins, which we'll come to in the next set of lectures. All of them are now present in these eukaryotes, you know, and this has happened at least with the actin and microtubules. You can see that there were versions of this with motor proteins, for example. Boom, right? It's there. Now, it's possible that there is a version of motor proteins that is present in those prokaryotes that we don't know of. I mean, till 20 years ago, we did not think that there was an FTSZ, right, in prokaryotes. So it's possible that this is just waiting to be discovered, and this table will change, and we will start seeing things here as well as we understand this better. But this is just to give you a flavor of what this would have looked like in prokaryotes. Again, the architecture of this tubulin strand, for example, and it's assembled in very distinct structures, right? So it's not the same architecture that you know of in mammalian cells, but in kind of fourth generation and making a ring to kind of divide bacteria, the FTSZ ring, you know, is thought to play a very important role, right? So I'm going to stop here.